Water. It's in the cells inside of us and fills our ears and toes. Yes, 2/3 of all of us is just some tiny H2Os. 2/3 of the surface of planet Earth is covered with water, and about 2/3 the weight of a human being is water. Coincidence? Not really. Life on Earth almost certainly began in water, and so far as we know, liquid water is an irreplaceable requirement for all life. Fresh water is the form we need. Salt water won't do, and it needs to be reasonably free of heavy metal ions, as Flint, Michigan can attest. Water is uniquely valuable because it has some special and peculiar properties that make it different from all other liquids in ways that are crucial to the processes that support life. Unlike most liquids, as water freezes, it expands, thus reducing its density. Very few compounds have this property. It is why ice floats in liquid water and why fish can live through winters in lakes in cold environments. That water is a liquid across a wide range of temperatures we consider normal is in fact quite unusual too. If water mimicked a similarly sized molecule like methane, water would become a gas at or below room temperature. Methane itself boils to gaseous forms at temperatures far below the coldest winter day. If water weren't a liquid, then we would have to pass because there's no way a cell could be comprised of gas. Its membranes all would come apart, the insides all askew. No nucleus, no organelles, and chaos would ensue. The key to the behavior of water is in a property of atoms called electronegativity. Electronegativity is a measure of the affinity of an atom for its nucleus, for its most outermost electrons. You can think of it as how strongly the nucleus of the atom holds onto those electrons. Atoms whose nuclei hold on tightly to outer electrons have high electronegativity values. Electronegativities of elements vary considerably. Fluorine and oxygen have the highest electronegativities. As we move from right to left across the periodic table, the electronegativity values tend to decrease. Nitrogen is lower than oxygen. Carbon is lower than nitrogen. Elements on the left side of the table, like sodium and potassium, have very low electronegativity values. The exception is hydrogen, but its single electron is also a special case. So why are there differences in electronegativity important? Well, consider a molecule of water made up of an oxygen atom and 2 hydrogen atoms. Oxygen has that very high electronegativity value of 3.44 compared to hydrogen's of 2.22. That means that the nucleus of an oxygen atom likes electrons more than does the nucleus of each hydrogen atom. So although oxygen can make a bond to share electrons with hydrogen, the oxygen does not share equally. We can think of oxygen as an electron bully, keeping shared electrons in each bond closer to itself and farther away from the hydrogens. Though this setup is still a covalent bond, in that the electrons are shared, the oxygen holds the bond electrons closer, so it acquires a partial negative charge. This is written delta minus, and since hydrogen is farther away from the bond electrons, the hydrogen gets a partial positive charge called delta plus. It is this non-uniform charge distribution that gives water its very interesting properties. When we look closely at the structure of a water molecule, the shape is a relaxed V, with the oxygen at the bottom of the letter and the hydrogens at the 2 points on top. That means the bottom of the V where the oxygen is will be slightly negative, and the tops of the V where the hydrogens are will be slightly positive. A molecule with an uneven distribution of charge like this is described as being polar. Each water molecule is in close proximity to several other water molecules, each with the same partial charge distributions. Opposite charges attract, so partial positive charges on the hydrogens of one water molecule are attracted to the partial negative charge on each oxygen of neighboring water molecules. Interactions arising between these partial charges are called hydrogen bonds. Though hydrogen bonds are possible between atoms of many molecules, we see them mostly involving oxygen, nitrogen, sulfur, and of course, hydrogen. And as we shall see, hydrogen bonds make the living world go round because their weakness is actually their strength. Two of the most important structures in our cells rely on hydrogen bonds for stability. First, the complex folded structure of proteins is stabilized by hydrogen bonds. Second, the double strands of DNA are held together nicely by hydrogen bonds. Hydrogen bonds are why water remains liquid at room temperature. These bonds are how water molecules attract and hold on to each other. Although such bonds are not permanent, each water molecule in liquid water is constantly making and breaking links with its neighbors in a sort of group dance, swapping partners but staying connected. Breaking all these bonds at the same time to turn water into steam takes a lot more heat than would be needed for molecules not held together by such bonds. Conversely, when temperatures drop and thermal motion is minimal, H2O molecules take a tiny step back, and each water molecule hydrogen bonds to 4 neighboring H2Os. This creates the highly ordered structure in an ice crystal, and because frozen water is in this crystal form, ice is less dense than water. Hydrogen bonds can also form within and between molecules. The same electronegativity difference that gives rise to the hydrogen bonding in water also allows amino acids to interact within proteins. Whenever large electronegativity differences exist between hydrogen and an atom, hydrogen bonds are possible. For example, hydrogen has much weaker electronegativity than nitrogen, 3.04, and forms hydrogen bonds when linked together. Carbon at 2.55, on the other hand, is much more similar to hydrogen, so it does not form hydrogen bonds to any significant extent. Every amino acid in every protein contains NH bonds and C double bond O bonds, and hydrogen bonds can form between those hydrogens of the NH and the oxygens of the C double bond O. Similarly, every nucleotide in every DNA or RNA contains those same bonds. And because most of the body is water, the hydrogen bonds within these biomolecules are constantly being invited to dance with water. That's because water is held together by these hydrogen bonds. Water is an effective medium and a solvent for molecules important for life, both inside cells and in the fluids that surround them. A solvent, of course, is a liquid that dissolves something. Ocean water is full of dissolved salts. Wells in rural India are contaminated with arsenic, and we've all learned how lead in pipes in Flint, Michigan made their water undrinkable. Water uses its partial charges to dissolve compounds. Table salt, or sodium chloride, can be ionized in water. Salt and electrolytes you see listed in drinks are held together by so-called ionic bonds between two atoms with very different electronegativities. Salt is a great example. Sodium's very low affinity for electrons, less than 1, and chlorine's very high affinity, over 3, means that sodium essentially gives up its electron to chlorine when they bond. Sodium then has a net charge that's fully positive and is what we call a sodium ion. On the other side of the molecule, chlorine gains sodium's electron and is fully negatively charged, forming a chloride ion. Sodium's positive charge is strongly attracted to chloride's negative charge, creating what is called an ionic bond. When NaCl dissolves in water, the partial positive charges of water's hydrogens surround the negative charges of the chlorides, while the partial negative charges of water's oxygens surround the positive charges of the sodium plus ions. As a result, the ions of sodium chloride get pulled apart from each other by water molecules, and salt dissolves in water. The same is true for any molecule with ionic bonds. And some of the resulting ions are very important for cells. For example, sodium ions and potassium ions are critical for the signaling of nerve cells, while chloride ions provide important counterions to balance charges. Many organic compounds, by contrast, such as sugar, dissolve in water because the partial charges of their own hydrogen bonds can mingle directly with the hydrogen bonds of water. No need to ionize. Glucose is a typical sugar containing numerous hydroxyl, that is OH groups. Glucose has the same uneven sharing of electrons between oxygen and hydrogen as water does. So just like water, the oxygens of the OH groups in glucose have partial negative charges, and their hydrogens have partial positive charges. Water's partial charges can dance with the partial charges of glucose to pull individual sugar molecules into the solution. But other molecules like oil and fat do not dissolve or mix in water. And this is because they have long chains of CH bonds that do not ionize and do not form hydrogen bonds. The electronegativities of H and C are not identical, but they're similar enough that the electrons in these bonds are pretty equally shared. The charge distribution is effectively uniform, and the bond is effectively nonpolar. So fats can neither ionize like salt nor readily form hydrogen bonds like sugar. And because fats have neither ions nor hydrogen bonds, water can't pull fat molecules apart from each other. Molecules like fats that do not dissolve in water are called hydrophobic, a term that means water-fearing. Molecules that do dissolve in water are called hydrophilic, or water-loving. Not all molecules fall neatly into one of these two categories. Some molecules are ambivalent about water. They have a sort of inner conflict. Part of them likes water, while another part avoids water. These molecules are amphiphilic. Soap is a simple example of an amphiphilic compound. It is partly hydrophilic and partly hydrophobic. The hydrophilic end interacts with water readily. The hydrophobic end avoids interacting with water. You might guess an amphiphilic molecule would have an intermediate level of solubility in water compared to hydrophilic and hydrophobic compounds, but it's more interesting than that. Like a clique of middle school kids huddling with each other and rejecting those who are different, the hydrophobic regions of amphiphilic molecules, like soap, arrange themselves to be near each other and away from water. Meanwhile, their hydrophilic ends happily associate with water molecules. All this combines to form a spherical, bubble-like structure called a micelle. This micellar structure is critical to how soap removes grease when we wash our hands or do the dishes. Grease contains nonpolar hydrophobic molecules that don't make hydrogen bonds and don't like interacting with water. So how do you wash the grease away with water? Soap micelles in water have nonpolar tails that interact with grease droplets and start to pull them apart as they shield them from water. Meanwhile, the hydrophilic ends of the soap interact happily with water. The soap micelle is effectively a carrier of the grease. With this action, the grease is dissipated. We say it is solubilized, much like the salt was earlier. This chemical property of nonpolar parts of molecules to associate with each other to the exclusion of water is called the hydrophobic effect, and it turns out to be very important for forming cellular membranes. Proteins also rely on the hydrophobic effect to hide hydrophobic parts away from water in a process called folding, as we shall see in a later lecture. Okay, let's review. Electronegativity differences between hydrogen and oxygen make water a polar molecule. Other polar molecules interact readily with and dissolve in water. We call them hydrophilic. Nonpolar molecules avoid water, so we call them hydrophobic. Molecules like soap are part hydrophobic and part hydrophilic, and we call them amphiphilic. Soaps facilitate dissolving and transport of hydrophobic substances in water. We'll see a similar strategy employed by proteins for moving fat and cholesterol in the bloodstream in a later lecture. Another property of water important in biological chemistry is water's own ability to ionize or break into ions. This happens only to a very small number of water molecules. About one water molecule in more than 500 million will ionize, meaning it's less likely to happen than a single Powerball ticket winning the big prize. But it does occur, and it is important. When water ionizes, it breaks into positively charged hydrogen ion, H+, called a proton, and a negatively charged hydroxide ion, OH-. The process results in the formation of ions because oxygen's higher electronegativity means it takes the unequally shared electron entirely for itself when the split occurs, leaving the positively charged proton with no electron at all. With these conditions, water has gone from being H2O with no net charge to H+ and OH-, each of which has a charge. A fundamental principle in chemistry is that any substance that can donate a proton is called an acid, while one that can accept a proton is called a base. Looking at an ionized water molecule becomes apparent that in addition to its other peculiarities, water acts as both an acid and a base. When it ionizes, it releases a proton acting as an acid, but it simultaneously releases a hydroxide ion which can accept a proton to act as a base. Now, water is not a very strong acid base because only a minuscule amount of it ionizes. Most of it remains intact water molecules. The proton joins to a water molecule making H3O+. In pure water, the concentration of H3O+ and hydroxide ions is equal, with each being about 1 × 10⁻⁷ molar. So the solution remains neutral overall. The fact that the concentration of protons, that is hydrogen ions, in pure water is about 1 × 10⁻⁷ molar is a basis for the pH system to measure the acidity of a solution, where the H in pH stands for the hydrogen proton. pH is defined as the negative logarithm of the proton concentration. For pure water, the concentration is 1 × 10⁻⁷ molar. The negative logarithm of 1 × 10⁻⁷ is 7. So a neutral solution, which is neither acidic nor basic, is said to have a pH of 7. If the hydrogen ion concentration is increased to 1 × 10⁻⁶ molar by the addition of an acid, the pH goes down to 6. So you notice that each change of 1 pH unit reflects a tenfold increase in proton concentration, from 1 × 10⁻⁷ molar to 1 × 10⁻⁶ molar. Now you might be wondering what all this has to do with biochemistry. And the answer is that everything, all cellular processes, involve water. So the pH of cellular and extracellular fluids plays an enormous role in biological chemistry. The hydrangea, for example, is a very visual example of the effects pH change can have on an organism. Hydrangeas grown in acid soils have a blue color, whereas those grown in more alkaline soils are pink. Litmus paper pH indicators use the same principle but with the dye colors reversed, changing from an acidic pink to a basic blue over a pH range from about 4.5 to about 8.3. The pH of the cytoplasm of cells has to remain within a much narrower pH range for cellular reactions to occur appropriately. Likewise, the pH of our bloodstream is confined to a very narrow range of values, almost always between 7.35 and 7.45. If the pH should rise above 7.45, a condition known as alkalosis occurs. And if it gets below 7.35, then acidosis occurs. Alkalosis affects the nervous system, heart muscle, and liver, while acidosis has other undesirable effects ranging in severity from headaches and drowsiness to coma. If the blood pH drops below 6.8 or rises above 7.8, bonds holding together critical proteins can get destroyed and death results. Fortunately, there are buffering mechanisms in our bodies to keep pH within that narrow range and avoid catastrophic consequences. To understand buffering mechanisms, we must first understand buffers. Weak acids act as buffering agents, so we'll examine the behavior of one weak acid, acetic acid, to help understand how they work to keep the pH constant. Acetic acid has a vinegary taste. It typically makes up about 5% of the volume of vinegar in the store. Anyone who's ever had a wine go bad knows acetic acid. Remember that a weak acid is one that does not ionize readily. It does not give up protons easily to water. Strong acids like hydrochloric acid, HCl, by contrast, ionize completely. For every million molecules of HCl put into water, 1 million protons and 1 million chloride ions will be produced, leaving no unionized HCl behind. Similarly, there's a strong base called sodium hydroxide, or NaOH, which also completely ionizes in water, where it forms sodium ions, Na+, and hydroxide ions, OH-. When we do the same thing with a weak acid, only some of it ionizes. With that in mind, let's take a look at what happens to our weak acid, acetic acid, if we try to perturb the pH of the aqueous solution it is in by adding a strong base like NaOH to it. Our graph shows how much NaOH we've added on the horizontal axis, and the resulting pH is on the vertical axis. Ready? This is called a titration plot, and we begin with just acetic acid. We start our plot at the maximally acidic pH of 0, and then we start adding a strong base, NaOH, to it. The OH- hydroxyls from the base react with the protons in the solution, causing them to form water, thus reducing the concentration of protons with each addition of NaOH. But if we keep adding more and more NaOH, which should keep increasing the pH, an interesting thing happens. We see that adding more NaOH after a certain point doesn't change the pH much. It rises much more slowly in proportion to the amount of NaOH added. Finally, after we put in enough NaOH to push the pH up beyond about pH 6.26, the system goes back to quickly being responsive to the addition of more base. The graph shows that there is a range of pH values, roughly 3.26 to 6.26, over which the system stubbornly resists change. This resistance to change in pH is called buffering, and it's typical over a range of about plus or minus 1.5 units of the pH from the midpoint of the resistance. Now, what exactly is happening here? When we add NaOH, hydroxide ions combine with the protons from the acetic acid, and we make water. And when we leave what is called a salt or conjugate base of CH3COO- or an acetate ion in solution. We commonly use the word salt to describe what remains after an acid loses its proton. That is at least partly because the missing proton could be replaced by another ion like sodium, and that comes from salt. The more NaOH strong base we add, the more protons are given up by acetic acid, and the protons combine with the OH groups from NaOH to form water. This continues until all the acetic acid is converted to the CH3COO- acetate salt. If we note the amount of NaOH we added to completely deprotonate the acetic acid, we work back to a relatively stable balancing point, an inflection point, if you will, at which exactly half of the acetic acid molecules have lost their protons and the other half have not. The pH for the inflection point is where the acid and conjugate base are in balance, and it's called the pKa. The stronger an acid, the lower the pH will be for its pKa balancing point. Each acid has its own relatively stable pKa inflection point, as well as its own range above and below that value over which the buffering takes place. For acetic acid, that pH that corresponds to the pKa is 4.76. For lactic acid, which is produced in our muscles when we overexercise, the pKa inflection point is 3.86. Other acids have 2 or even 3 carboxyl groups, or other groups that can donate a proton. Such acids, which include all the amino acids, have a distinct pKa value for each of their ionizable groups. Carbonic acid, for example, has 2 groups, one at 6.37 and a second at 10.2. A more general relationship between pH and pKa is expressed in the famous Henderson-Hasselbalch equation, which relates the pH of a solution to the always constant pKa value, plus an adjustment for the actual amount of salt and weak acid present. The adjustment is a ratio of the concentrations of conjugate base over weak acid because increases in pH produce more conjugate base. And it goes the other way. Increases in conjugate base also increase pH. And we take a logarithm of that ratio because pH and pKa are already logarithms. So to summarize, different weak acids have different pHs at which half of their carboxyls are protonated. And this specific pH for a given acid is called its pKa. We see that when we perturb the pH around a weak acid, there's a range of pH values on either side of the pKa within which there is a stubborn reluctance to change called buffering. Buffering, or resistance to change in pH, is enormously important in living organisms, which have to maintain the pH of cells and extracellular fluids within very, very narrow ranges. So how do our bodies maintain the pH within such a narrow pH range? The weak acid of the blood's buffering system is formed when carbon dioxide produced by tissues combines with water to make H2CO3, also known as carbonic acid. Carbonic acid has two pKa values, but its lower one of 6.37 is the more relevant one biologically, being within 1.5 pH units of blood's pH of 7.4. It readily ionizes to produce a mix of conjugate base and weak acid at this point. Maintenance of the blood pH at the value of 7.4 requires the ratio of carbonic acid to bicarbonate ion be maintained at a 1:20 ratio. The carbonic acid is a proton donor, so the protons it releases are bound to the hemoglobin in blood, which simultaneously releases oxygen to the tissues. The ongoing removal of protons by hemoglobin favors continued dissociation of carbonic acid and maintenance of the 1:20 ratio of acid to salt. When the blood reaches the lungs where the oxygen levels are high, hemoglobin releases the bound protons as it binds oxygen instead. The protons released then bind to conjugate base, HCO3-, which tips the balance back in the favor of the acid, H2CO3. An enzyme in the lungs then converts the carbonic acid, the H2CO3, back to water and carbon dioxide, which is removed from the lungs through exhalation. Carbon dioxide is one product of respiration, but another important one is water. There are about 100 grams of water produced per 100 grams of fat, 60 grams per 100 grams of carbohydrate, and 42 grams of water per 100 grams of protein. For some organisms, such as migratory birds and animals living in the desert, metabolic water may sometimes be their only source of hydration. Incidentally, on the subject of pH, there is a nutritional fad to be aware of. It involves whether what you eat affects your blood pH. The answer appears to be no. There is no evidence that diets such as the so-called alkaline diet, which shuns meats, cheeses, and grains in favor of fruits and vegetables, can do anything to the pH of your blood or cells. While pretty much everyone agrees that fruits and vegetables are excellent nutritional components of a healthy diet, claims that they change the pH of cells or blood has no biochemical basis. Remember, the body's buffering system holds blood pH in a very narrow range between 7.35 and 7.45. In fact, if a fad diet did change blood pH, it almost certainly would be detrimental, not beneficial. Your body's buffering systems are already keeping your pH where it needs to be. Well, let's wrap this up. Everybody knows that water is important, but let's remember why. Roughly 2/3 of the body's mass is water. Water is not like other small molecules and affects the behavior of all other biological molecules. Water is a polar molecule thanks to oxygen bullying a couple of hydrogens for a bigger share of their bond electrons. And because polar molecules can form hydrogen bonds, water's V-shaped molecules bond with each other and with other charged or partially charged molecules. And when weak acids dissociate in water to form weakly basic salts, the weak acid and conjugate base provide buffering that is important for cells to resist pH changes. So remember, there are a lot of implications arising from the simple hydrogen bonds of water that will open doors to much bigger topics later. Though it is the covalent bonds that stick us together, it is water, through the effects of hydrogen bonds and pH on the structure and activity of macromolecules, that make everything play nice together.